The traditional way of viewing the origin of life is through a three-domain theory. This theory involves the evolution of bacteria and archaea around 3.5 billion years ago. Eukarya evolved around 2.1 to 2.4 billion years ago. Eukaryotic genes for nuclear proteins are more similar to archaeal counterparts, but that eukaryotic genes for energy metabolism and lipid biosynthesis more closely resemble bacterial genes. This leads to the hypothesis that eukaryotes evolved from an ancient endosymbiosis or cell fusion event between archaeon and bacteria. Recently, evidence has been revealed that supports a hypothesis in which there are only two domains of life, bacteria and archaea. This hypothesis is known as the eocyte hypothesis, in which the origin of eukarya evolved from the archaea domain and proposes a sister group relationship with the eocytes. In looking at the phylogenetic trees of these two hypotheses, it can be seen that in the traditional tree of life, the archaea forms a monophyletic group, a group consisting of the ancestor and all of its descendants, whereas the eocyte hypothesis suggests a paraphyletic grouping of the archaea, in which the eukaryotes are contained within archaea. The determination of a 2 or 3 domain origin of life is a difficult process. One of the challenges is due to the large amount of time that has passed since the origin of domains. In that time, there has been an accumulation of substitutions in DNA and protein sequences that have erased signals of a relationship between eukarya and archaea. There are only a finite number of genes that occur on slow-reacting sites that can be followed from the origin of life. Another problem in early tree of life construction is long branch attraction. LBA causes species to seem more closely related than they really are due to mutations or traits occurring independently. The third major challenge in constructing relationships was that the trees were constructed using unrealistic assumptions in the parsimony and distance methods used. Studies have sought to mitigate these challenges. One such way was improved sampling of genetic sequences in which sequences from uncultured prokaryotes can be obtained. These improvements have allowed scientists to break up longer branches and discover relatives of the eocytes, Chorarchaeota, Thaumarchaeota, and Agarchaeota. Together with these eocytes, they make up a group called TAC, which are theorized to be the closest relatives to the eukaryotes. If this is correct, the most important place to look for prokaryotic homologs is along the TAC group. Members of TAC have genes for actin and tubulin, which are essential components of the eukaryotic cytoskeleton. One of the common challenges to the eocyte hypothesis is the reversion of the archaeal host membrane back to a bacterial-type plasma membrane. The plasma membrane of the eukaryotes and bacteria are very similar in that they both contain phospholipids in which fatty acids are covalently bound to the SN-glycerol 3-phosphate via an ester linkage. In contrast, archaea contain phospholipids with isoprenoid chains linked to SN-glycerol 1-phosphate via an ether bond. This is most easily explained using a 3-domain hypothesis in which the eukarya and bacteria have a shared common ancestor. Although this is true, there has been similarities in the genes needed for synthesis of both types of lipids found in all 3 domains. Also, there have been experiments in which bacterial and archaea membranes have been combined to create stable artificial membranes. Considering this, the similarities seen between bacteria and eukarya can be explained by the reversion of archaeal membranes to bacteria-like membrane. The original 3-domain theory hypothesized modern eukaryotes descend from lineages that never had mitochondria, meaning that mitochondria came after nuclei. New evidence shows that some eukaryotes possess mitochondrial homologs, hydrogenosome or mitosome, implying that mitochondria were obtained before radiation of the eukaryotes. This is strong evidence that the origin of eukaryotes comes after the origin of bacterial group. Phylogenetic trees allow us to make connections and view the ancient past, but they are difficult to build and interpret. Movements in data and methods have helped us to give more insight to these phylogenies. Eukaryotes appear to have genes through gene transfer from different bacteria, including mitochondria and plastids, but the core set of proteins are more similar to archaea. This evidence of horizontal gene transfer makes it so no one tree is sufficient to give the origin of the modern eukaryotic genes. But the eocyte tree is the best supported when viewing the origin of genes related to translation and are resistant to HGT. If correct, the eocyte hypothesis suggests that TAC might contain the clues to the origin of complex eukaryotic structures. The takeaway is the eocyte hypothesis suggests 2 fundamental domains of life, the archaea and bacteria, and the eukarya is a subgroup of archaea, but much is left to be revealed about the origin of the eukarya.